Hello guys, welcome to another video tutorial in Mushrooming Your Knowledge. Today I am going to discuss about the remaining part of prokaryotic DNA replication. In my previous video I discussed about the initiation process. The link is provided in our description box or you guys can click on the I button. Before starting the replication elongation phase, we have to know about some proteins which are involved in initiation phase. Let's discuss briefly about those protein. The first one is DNA protein. Let's take the pen. Okay, the first one is DNA A protein. Okay, DNA A protein binds to DNA A boxes within the origin to initiate DNA replication. So DNA A help to initiate DNA replication. The second one is DNA C protein. DNA C aids DNA A in the recruitment of DNA helices to the region. That means DNA C help DNA A to recognize the region that's where DNA helicase binds. The third one is DNA helicase or DNA-B protein. Separate double-stranded DNA. DNA-B separate double-stranded DNA-B, it goes in this direction and separate the double strand. It breaks the bases between two strands. It has 6 subunit or and it forms a ring-like structure. And it migrates to the 5' to 3' direction. The fourth one is topoisomerase or guidase. When DNA helicase is to break the hydrogen bond between base pairs and thereby unwind the strand, this action generates positive supercoiling ahead of each replication forks. We know that the bacteria or prokaryotes have the circular DNA. When here the replication bubbles from here and there from the two supercoiling, DNA topoisomerase or guidase help to remove the supercoiling. Here type 2 topoisomerase help to remove the positive supercoiling. And the fifth protein is the SSP protein that help to rewind the break DNA strand which break by the help of helicase. Here it binds SSB protein, that is the single stranded binding protein. It binds to the strand of parental DNA and prevent them from reforming a double helix. In this way, the base within the parental strand are kept in an exposed condition that enables them to hydrogen bond with individual nucleotide. When helicase breaks those strands, there is a possibility of rejoining of them. So SSB binds here and there and prevent them from rejoining. So today we are going to discuss about elongation and termination. As I told you guys, replication, the whole process don't have any stages. We divided all those just for our better understanding. The first one is initiation, which I already discussed, that means starting. Second one is elongation and third one is termination. Elongation means growing and termination means ending. So today we are going to discuss about elongation and termination. So let's start. The next event of DNA replication is elongation. Just wait a minute. Elongation. Today the first one is elongation we are going to discuss. It involves the synthesis of short strand of RNA rather than DNA called RNA primers. So primers means prime, that means helps to start, and the first RNA primers are formed in DNA replication. Just mind it, okay? RNA primers are formed in DNA replication, not DNA primers. Those are the RNA primers. These strands of RNA are synthesized by the linkage of ribonucleotides via an enzyme known as primase. This enzyme synthesis short strand of RNA of 10 to 12 nucleotides in length. Okay, 10 to 12 nucleotide lengths of a RNA primer. This short RNA strand start or prime the process of DNA replication. So, the length of 10 to 12 nucleotide RNA primers help to start the synthesis of DNA replication. In the leading strand, there is two strands. The first one is leading strand and the second one is lagging strand. The leading strand, a single primer is made at the origin of replication, and in the lagging strand there are multiple primers are made. Then the RNA primers are eventually removed. After they complete their work, the primers are removed from this leading or lagging strand. So in the leading strand, a short piece of RNA called primer, that is binds in the bind here at the ending, formed by the help of primase enzyme, comes along and bind to the end of the leading strand. Here I indicate the binds to the end of the leading strand. The primer act as the starting point of DNA synthesis. Here the primer act as the starting point of DNA synthesis. DNA polymerase bind to the leading strand and then walks along it. That means walk along in this direction. Here helicase breaks the double strand, and the primers are going in this direction, and the new and synthesis of new strand are formed in this direction. So adding new complementary nucleotide bases AGCT, that means the ATGC, those are the adenine, guanine, thymine, cytosine, to the strand of DNA in the 5' to 3' direction. That is the 5' to the 3' direction. If it is the 5', then here it is the 3'. We can say that 3'. This sort of replication is called continuous. So leading strand is the continuous strand, and the other one is lagging strand. Numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. And here it is the lagging strand. Here the first primase, the second primase, and so on. If the strand is growing, growing in this direction, and the third one or fourth one is adding. So the chunks of DNA called Okazaki fragment are then added to the lagging strand also in the 5' to 3' direction. Those fragments known as Okazaki fragments. This type of replication is called discontinuous as the Okazaki fragment. Those fragments, that means the separated parts. So it is discontinuous, and the leading strand is a continuous strand. You guys can see that it is a continuous strand, and the lagging strand, it is discontinuous. One fragment, the second fragment, and so on. A type of enzyme known as DNA polymerase, or the abbreviation is known as DNA pol. Okay, DNA polymerase, we can say also the DNA pol, that one, DNA pol, that part, is responsible for synthesizing the DNA of the leading and lagging strand. So DNA pol help to RNA primers help to start the process, and DNA pol help to synthesize the new DNA strand. This enzyme catalyzes the formation of covalent bond between adjacent nucleotides and thereby makes the new daughter strand. In E. coli, 5 distinct proteins functions as DNA pol and are designated polymerase 1, 2, 3, 4, and 5. So the first part is we can divide those 5 types into two different part. The first one is the DNA pol 1 and 3 are involved in normal DNA replication. It helps to synthesize the new strand. New strand is formed by the help of 1 and 2. Pol 1 and 3. And the pol 2, 4, and 5 play a role in DNA repair and replication of damaged DNA. So if a thing exists, it will damaged by something. So DNA pol 2, 4, and 5 help to repair DNA replication, and DNA 1 and 3 help to synthesis the new DNA strand. Okay, just mind it or note it down that 1 and 3 help to synthesize the new one, and the 2, 4, and 5 help to repair the DNA replication. As researchers began to unravel the function of DNA polymerase, two features seemed unusual. Okay, note this point. Two features seemed unusual. DNA polymerase cannot begin DNA synthesis. You guys can know, I told you previously that DNA polymerase can synthesis or repair the DNA strand, but RNA primers help to initiate the replication. Rather, this type of enzyme can elongate. DNA poly only help to elongate only a pre-existing strand starting with an RNA primers or existing DNA strand. That means 1 and 3 help to start elongation in a pre-existing strand, or RNA primer help to DNA poly 2, 4 and 5 help to repair the existing strand. A second unusual feature is the direction of strand synthesis. DNA polymerase can attach nucleotide only 5' to 3' direction, not in 3' to 5' direction. Okay, remember 5' to 3' direction, not, mark that point, not in the 3' to 5' direction. In the second paragraph, due to those unusual features, the synthesis of the leading and lagging strands show distinctive differences. The synthesis of RNA primers by primase allows DNA pol 3 to begin the synthesis of complementary daughter strand of DNA. Daughter strand, what is daughter strand, what is template strand, what is parent strand, I discussed in my previous video. You guys can see that the link is provided in our description box. DNA pol 3 catalyzes the attachment of nucleotide to the 3' end of each primer in the 5' to 3' direction. Okay, not in the 3' to 5' direction. Okay, not in the 3' to 5' direction. In the leading strand, one RNA primer is made at the origins. In the leading strand, there is only one RNA primer, and in the lagging strand there are many primers. And then DNA pol 3 can attach nucleotides in the 5' to 3' direction as it slides toward the opening of the replication fork. So DNA pol 3 help to attach new DNTPs, that is the AT or GC, whatever it is, in the complementary sequence. The synthesis of the leading strand is therefore continuous. So DNA pol 3 is responsible. DNA pol 3 polymerase 3 is responsible for the most of DNA replication. So DNA pol 3 is mainly responsible for DNA replication. It is a large enzyme consisting of 10 different subunits. Okay, remember 10 different subunits. 10 different subunits that play various role in the DNA replication process. There will be a full stop. The alpha subunit actually catalyze the bond formation between adjacent nucleotides, and then the remaining 9 subunits fulfill the other functions. So alpha subunit, what does alpha subunit do? Actually catalyze the bond formation between adjacent nucleotide, and the remaining 9 subunits fulfill the other functions. The complex of 10 subunits together called DNA pol 3 holoenzyme. So DNA pol 3 have 10 subunits. I will make another video in and describe the structure of DNA pol 3, or you guys can say that DNA. I will discuss all the DNA polymerase in that video. The catalytic subunit of all DNA polymerase has a structure that resembles a human hand. Okay, the structure of DNA pol 3 is also resemble of a human hand, as shown in this figure. The template in this figure, there's the figure. The template DNA is threaded through the palm domain. What is the palm domain? That's the palm domain, that's the thumb domain, and that's the finger domain. Through the palm of the hand, the thumb and finger are wrapped around the DNA. The incoming DNTPs, that is the D stands for deoxyribo, N stands for nucleoside, T stands for tri, and the PS stands for phosphatase, that is the deoxyribonucleoside triphosphates, enter the catalytic site, bind the template strand according to AT-GC rule, and then their covalently attached to the 3' direction of the growing strand. Here the 3' direction and the 3' direction, and it is the 3' direction. It start to adding, and the here the it is first add, then this one, and going on. Okay, so DNA pol 3 helped to addition of new DNTPs as per ATGC rule. So in the next slide, in the lagging strand, the synthesis of DNA also elongate in the 5' to 3' manner. Okay, 5' to 3' manner in the lagging strand. Previously we discussed about the leading strand. Okay, and in this one we are going to discuss about the lagging strand. Wait a minute. But it does so in the direction away from the replication fork. Away from the replication, that was this is on the lagging strand, that was the lagging strand. This one in the direction of the replication fork, and that was away from the replication fork. In lagging strand, RNA primers must repeatedly initiate the synthesis of short segment of DNA. Thus the synthesis has to be discontinuous. It forms in this direction. So helicase cut, break the strand, the single strand will be formed, and the RNA primer, here the primer binds, and the DNA pol 3 help to synthesis the new strand, and this process is going on. The length of these fragments in bacteria is typically 1000 to 2000 nucleotides. In eukaryotes, the fragments are shorter, 100 to 200 nucleotides. Each fragment contains a short RNA primer at the 5' end. That is a 5' end. Contain a RNA primer at the 5' end. Okay. 5' end, which is made by primase. So primase help to form the RNA primer. The remainder of the fragment is a strand of DNA by DNA pol 3, and DNA pol 3 help to form this strand or this strand or this one. The DNA fragments in this manner are known as Okazaki fragment after Reiji and Tuneko Okazaki. So mark the name of those scientists. First one is Reiji and second one is Tuneko, who initially discovered them in the late 1960s. Okay. To complete the synthesis of Okazaki fragments within the lagging strand, 3 additional events must occur. So the first one is the removal of RNA primers. They complete their work, RNA primers complete their work, and they are removed from the system. The second one is synthesis of DNA in the area where the RNA primers have been removed. So RNA primers removed, then this place is blank. So there must be synthesis of new DNA strand, or those strands are not joined in a single strand and not formed a single strand. The third one is the covalent attachment of adjacent fragment of DNA. The fragments of DNA must join. So unequally, the RNA primers are removed by the action of DNA pol 1. Here it comes DNA pol 1. We discussed about the function of DNA pol 3 in our previous slide. So DNA pol 1 help to remove the RNA primers. Okay, this enzyme has 5' to 3' exonuclease. Exo means outer. Okay, outer part, exonuclease activity, which means that DNA pol 1 digest away the RNA primers in a 5' to 3' direction, leaving a vacant area. So primers are removed and this area will be vacant. DNA pol 1 then synthesize DNA to fill this region. DNA pol 1 synthesize the new strand of DNA, and it will help to fill up the vacant area. It uses the 3' end of an adjacent Okazaki fragment as a primer. Okay, so replication is terminated when the replication fork meet at the termination sequence, that is termination, the third stage. On the opposite side of the E. coli chromosome, that is a circular chromosome, you can see that it is a circular chromosome from the odyssey. opposite side of Odyssey, that is the TAR sequence. Okay, TAR means termination, that means ending of the termination sequence, called TAR sequence. Okay, TAR sequence. A protein known as termination utilization substance, that is the TAS. T for termination, U for utilization, and S for substance, bind to the TAR sequence and stops the movement of the replication fork. So TAR-TAS complex will be formed and it will stop the replication. So TAR sequence is formed opposite to the Odyssey site. So as shown in this figure, one of the TAR sequence digested T1 prevent the advancement of the fork moving left to right. Just sorry, pin that here's the T1 or TAR1 sequence, and here's the TAR2 or termination 2 sequence, TAR2, and this one the TAS. T1 prevent the advancement of the replication moving left to right. Okay, TAR1, that is the TAR1 moving left to right, but allows the movement of the other fork. Alternatively, T2 prevent the, not the T2 mama. Okay, Vivica vines, not Vivica vines, it's replication, not the T2 mama of Vivica vines, it's T2. Okay, T2 prevents the advancement of the fork moving right to left. Okay, moving right to left, and the TAR1 is left to right, but allows the advancement of the other fork. In any given cell, only one TAR sequence is required to stop the advancement of one replication fork. If one TAR sequence is present, that's enough to stop the replication, and then the other fork ends its synthesis of DNA when it reaches the halted replication fork. In other words, DNA replication ends when opposite advancing forks meet. So DNA replication ends here, usually at T1 and T2. Finally, DNA ligase covalently links the two daughter strands. DNA ligase means addition. Ligase, not ligase means addition. Ligase help to add. DNA ligase is a protein help to add the strands, create two circular double-stranded molecule. Okay. So thank you. That's it for today. If you guys like our content and our videos, so like, comment, and subscribe our channel, and also share with your friends. Thank you.